Okay, we'll get started. Welcome everyone to WACBE webinars, our distinguished biomedical engineering webinar series hosted by the World Association for Chinese Biomedical Engineers, or WACBE. My name is Xinyu Zhang, or Zhang Xinyu in Chinese. I am from the Department of Biomedical Engineering at the Southern University of Science and Technology in Shenzhen, China. The format of our webinar is the following. Our speaker will present for approximately 40 minutes and will leave 15 to 20 minutes at the end for Q&A. Throughout the presentation, please feel free to use the Q&A feature at the bottom of your screen to submit your questions. We will try to get to as many questions as possible during the Q&A session. Today, we are excited to welcome Professor C.T. Lim from the National University of Singapore. Professor Lim is the NUS Society Professor and Director of the Institute for Health Innovation and Technology at the National University of Singapore. His research is in mechanobiology and microfluidics. He has co-authored over 400 peer-reviewed journal papers and given over 390 invited talks. He's an elected fellow of the IAMBE, Academy of Engineering Singapore, and the Singapore National Academy of Science. He has co-founded 6 spin-off companies. He and his team have garnered over 100 research awards and honors, including Highly Cited Researcher 2019, Asian Scientist 100 2016, Wall Street Journal Asian Innovation Award Gold 2012, and President's Technology Award 2011. Dr. Lim, welcome.
Okay, so first I want to thank Yasi for your very kind introduction. And also WACBE for inviting me to give this lecture. It's really an honor for me to give this distinguished biomedical engineering webinar. This is actually the second. Last week, the last one a couple of weeks ago was given by Professor Sylvia Wu and gave a very excellent lecture. I hope that I will not disappoint the audience for this talk. I also want to thank Edgar from Edgar and also Pimei for organizing this whole series of webinar, and I thought it's a very, a very important initiative because this is an opportunity for us to see how we can disseminate some of our research findings through such a very useful platform. For this talk, I'm going to focus on some of our work on mechanobiology of human diseases, and also to share with you how we actually use some of the findings for application in diagnosis and also therapy. This is the outline of my talk. I'll first talk about what is human disease mechanobiology, and I will actually cover 3 diseases. The first is on malaria, second on sepsis, and then I'll end with cancer. And these are the 3 diseases that we have been working very much in our lab, and we'll show you. I will show you how we go about studying some of these diseases, and then which leads to either the diagnosis or therapy. So when we talk about diseases, from some of our studies on certain diseases, we found that they do actually change in terms of mechanical, biomechanical properties, and this can include geometry and structure. And size, as well as deformability and cell adhesion. So some of the associated diseases are such as malaria, sickle cell anemia, spherocytosis, and also cancer. And in fact, some of the changes in this biomechanical properties can give rise to or arise on diseases. And we use the term mechanopathology to describe how changes in such mechanical properties can lead to pathological outcomes. So in my lab, we have been, although our focus very much is on the cellular level, actually we do also engage in quite a bit of the molecular and multicellular studies as well. And this diagram here shows some of the techniques we'll be using to conduct some of these studies. For example, the molecular level, we do use a lot of the AFM and magnetic force microscopy. And at the cellular level, actually, we do a lot of, for example, micropillars, where we put cells on the pillars to measure contractile forces, as well as microcontact multi-patterning to see how cells would respond to the surface that the cell adhered to, as well as developing other techniques like dual pipette assays and optical tweezers to measure cell adhesion, and microfluidics to be able to capture some of these disease cells from a large population or other cell types. At the multicellular level, actually, we've been also doing a lot of collective cell migration work in relation to, for example, wound healing, and a lot of some of the techniques we use also not only including microcontact printing, but also microfluidics, where we can flow cells, look at how cells will adhere to surfaces. So in our lab, basically, we conduct a lot of basic research for a start to understand the mechanobiology of human diseases. And the approach is somewhat actually quite of interest to our clinicians, because when we first started this research, the clinicians didn't quite understand what we are coming from. But after working in the last 10 to 15 years, I think they do appreciate us coming from the mechanobiological perspective. And this is a kind of a different approach in looking at diseases and how changes in mechanobiological properties can lead to the pathology of diseases. And as an engineer myself, I'm also very interested to see how this accumulated knowledge can be applied to develop very novel diagnostics and therapeutics. In fact, the first half is basic research, but the second half is a lot of translational work. So half of my group actually works a lot of basic research and the other half on translational work. And actually, this has led to a number of spin-off companies as well. So we were very keen to see how we can bring some of this research from the bench to the bedside. So in our lab, we really try to aim towards clinical relevance and also utility. So let me first go to the first disease, malaria. So this actually is the disease that I first worked on when I first started my lab. And it arises actually from a coffee break that I had with a colleague of mine from the microbiology department. So he was telling me that he's working on malaria, and he says that the cell, when a red blood cell gets infected, it becomes stiff and sticky, or the cell adhesion actually increases. And so when when he told me of this problem, I realized that actually it relates to mechanics, and we have some equipment that we can help them to study that, because when he was looking at this problem, they weren't able to quantitatively measure the stiffening of the cell or the increase in cell adhesion. But before I go into some of the findings, let me just introduce what is malaria. So malaria is actually one of the three deadliest infectious diseases. So it's a parasitic disease which is transmitted through a female Anopheles mosquito. So every year there are about 300 to 500 million clinical cases and 1 to 2 million deaths each year. So you can see that how serious this disease is. And how it works is this. So when an Anopheles mosquito take a blood meal from a person, it will inject parasites into the bloodstream, where the parasites will then go into the liver cells, and from liver cells it will develop into this merozoite, as you can see here. So once the merozoite goes in the bloodstream, it can actually attach to a blood cell, squeeze its way into the blood cell, causing massive changes to the blood cell. The blood cell becomes stiff and sticky. So if it becomes sticky, it can actually adhere to the surface of the blood vessel. And within 2 days, it will actually multiply from 1 to about 10 to 20. And thereafter, you will break out the infected cell and start to infect other red blood cells. And so this is the life cycle or the pathogenesis of the malaria. And so when we started this research, we also collaborated with a group, Professor Wusa, actually from the Mahidol University in Thailand. So we made a few trips to this place called Ratchathaburi, which is at the border between Myanmar and Thailand. So in there, over there, there's a village which housed about 500 villagers, and they're actually refugees from Myanmar. So when we were walking around the place, so this is the clinic where the villagers will come to seek help if they fall sick. And so when we walk around, there are a lot of adorable kids there. And when we look at, for example, rubbish, like for example, a discarded wheel like this, a wheel tire like this, and it's the perfect receptacle for collecting water. And we look into it, actually it's a breeding ground for mosquitoes. So the infection is pretty quite serious over there. So what happens is if a villager is sick, the villager will come to take a blood test. So the medical officer would do a finger prick to drop a few bloods onto a glass slide. And then after that, we'll do a Giemsa stain. And within half an hour, we will look underneath a microscope as shown here. So under the microscope, this is how it looks like. Let me show you an enlarged figure. So over here, you can see that these are actually, in the background, are actually red blood cells. They appear published in color is because of the Giemsa stain. And this larger patches of published color features are actually white blood cells. But if you look very closely, there are these tiny little specks inside the cell. These are actually malaria parasites. So from this picture itself or the image itself, you can tell that the person has been infected with malaria. So once we found that it's positive, the doctor or the medical officer will then administer anti-malaria drugs to the patients. So when we look at what happens to a red blood cell when it's infected with the parasite, it actually not only becomes stiffer, but also there's an increase in cell adhesion or cytoadherence. So this is what happens inside the red blood cell. So you can imagine from a red blood cell which just contains hemoglobin to one that contains many other other organelles and so on inside the cell apart from the parasite itself. And also on the surface of the infected red blood cell, you can see these tiny little knobs. These are actually proteins that gets embedded in the membrane of the red blood cell, and these are the knobs that cause the cell to be very sticky or very adhesive to the endothelium. And this is what causes this whole red blood cell to be both very stiff and very sticky. So if you look at a red blood cell, healthy red blood cell, this is how it looks like. So using AFM, we were able to image the red blood cell. You can look at the red blood cell itself is biconcave in shape, and on the surface is relatively smooth. If you were to look at the infected red blood cell, this is how it looks like. Where you look at the surface, there are all these little knobs. These are the cut protein that is secreted by the parasite that gets embedded in the membrane, and this is what causes the cell to be very sticky. And also, if you look at this red blood cell, right in the middle is actually where the parasite is. So it looks like a crater because when we adhere, when we smear the infected cell on the glass slide, the membrane actually sticks to the parasite. So over here, you can tell where the parasite is. So the mechanopathology of the malaria is such that when the cell becomes stiff and sticky, it will actually be able to clog the capillaries. And so this is bad news for our body because our red blood cell has to be very deformable and non-adhesive in order to flow through the circulatory system. But when it becomes stiff and sticky, this is what is going to happen. The capillary can get congested. And so this is a picture of a capillary that's blocked by the infected red blood cell from a cerebral malaria patient. So now that we have actually been doing quite a number of different studies, mechanobiological studies, not only probing the adhesion at the molecule level, but also performing a cell mechanics study at the cell level and thereafter multi-cell level, just to mimic what happens if a group of infected cells flows through the narrow capillaries. Due to constraint in time, I'll just talk about the cell mechanics work. So one of the first experiment we did was to use optical tweezer to stretch malaria-infected cells. So how the experiment works is this: we attach two beads to the side of the red blood cell. So for our case, we are using a single laser. So one bead is attached to the surface of the glass slide. The other bead is trapped using a laser. So when we move the laser or if you move the glass slide, we can actually be able to stretch the red blood cell. So let's look at the stretching of a healthy red blood cell. So this is what happens when we use the laser to trap one bead and move the laser, we can actually stretch the red blood cell. So over here, if you look at the experiment itself, the amount of force we apply is actually very small. It's about between 0 to about 170 to 180 piconewtons, but that's large enough to stretch the red blood cell. At the early stage of infection, you can see there's a parasite inside the red blood cell. The cell is still somewhat deformable. So this is about between 0 to 16 hours of infection. And as you can see, the cells is still quite deformable. Of course, there's already some increase in stiffness. But at the late stage, when we try to stretch the red blood cell, we can hardly stretch it. So this is really a very late stage infected red blood cell. So by using the same amount of force, we can hardly stretch it. So it shows how stiff the cell has become. We also did some finite element modeling so that we can work out what the increase in shear modulus is on the membrane. And from our studies, we found that the shear modulus obtained from optical tweezers experiment showed that from healthy to the late stage, there's about almost a 10-fold increase in the shear modulus of the infected red blood cell membrane. We also do other interesting experiments, like for example, micropipette aspiration of the infected cell. So if you look at this movie here, we use a very small pipette to aspirate the red blood cell. And you can see that the amount of cell that is sucked into the pipette is quite large. So that shows how deformable the cell is. At the ring stage, where you can see there's a parasite inside the cell, you can see that the cell is started to get stiffer. And then this is the trophozoite stage, which is the mid-stage infection. And then at the final stage, schizon, we can hardly aspirate the cell at all. You can see how stiff the cell has become. So from these experiments, we also wanted to see if we can develop some way of, and device that we can be able to, if we take blood from patient, pass through our device, we'll be able to harvest or be able to detect and obtain some of this infected red blood cells. So it's kind of a device that can go for enrichment and thereafter diagnosis. So we know that the number of red blood cells that's infected by malaria is actually very few. It's only a fraction of a percentage of the red blood cell, and they exist in a huge population of other healthy red blood cells. But we do know that there's a difference in biophysical properties, and as I shown you just now, there's a difference in terms of the deformability of the infected red blood cell versus healthy red blood cell. So using the differences in this cell deformability, we think that we can use this as a biomarker for us to enrich and diagnose these malaria-infected cells. So when we move ahead, we started to look at cell margination to see if we can make use of this phenomenon to isolate infected red blood cell. So we do know that when the red blood cell undergoes migration in a channel like this, or in our blood capillaries or blood vessel, there is this pulsar flow effect where the larger leukocyte actually gets marginated to the side of the wall of the channel or the blood vessel. So we know white blood cell is stiffer than red blood cell. And so if the infected red blood cell is also stiffer, then we might be able to use this cell margination effect to actually push the cell to the side of the wall and thereafter isolate them. So we came up with a microfluidic device like this, a very simple, very long channel microfluidic device. And it's about 3 cm long, but the cross-section is about 15 microns by 15 microns. And so the idea is that we can flow a red blood cell and infected red blood cell. And as it passes through, as it flows through this long channel, the stiffer red blood cell will get pushed to the cell channel, and thereafter we can retrieve them. So we are using cell stiffness here as a biomarker for us to separate the infected cells from the healthy red blood cells. So let me just show you a movie of what happens when we have an infected red blood cell flowing among a large population of healthy red blood cells. So you can see here the stained cell is an infected cell, and this is in hematocrit 40 to 50%, which is what we are seeing in our human body. as the infected cell flows through the channel, you can see that it gradually starts to marginate to the side of the channel. So using from this experiment, we know that the stiffer red blood cell actually will marginate to the side of the channel. So thereafter, we came out with this device to see if we can isolate them. So we do know as it flows through, the red blood cell, infected red blood cell will marginate to the side of the channel. So what happens is, as it comes out over at this region here, you'll see that a lot of the infected red blood cell will start to marginate to the side of the channel, and thereafter we will be able to try to isolate them. So this is midway, and then at the end of this device, where we a lot more of the infected red blood cell will start to go into outlet 1 and outlet 2. So this is what happened. So now you can see a lot of the infected red blood cell starts to marginate to the side channel, and this is one way where we can isolate the disease cells, infected red blood cells, and then send it for diagnosis. One reason why we want to develop this is also because sometimes when we do a finger prick and we have a drop of blood on the glass slide, sometimes it's actually not very easy to see the infected red blood cell, and sometimes because the smear itself has multiple layers of the cell, and to look under the microscope is actually quite challenging. And so if there's a way for us to take a drop or 2 drops of blood from a patient, pass through a device, and be able to separate the infected cells, then we have a more enriched infected red blood cell for us to look at under the microscope. So this is for malaria. The next disease I want to go to is actually sepsis. So we know sepsis is an adverse systemic inflammatory response due to microbial infection in blood. And this is actually, sepsis is actually a very serious problem because it's a primary cause of death in the ICU. And in fact, the mortality rate can range from about 28 to 50%. And also because of the antimicrobial resistance, a lot of these bacteria are now pretty resistant to drugs. So it's getting very hard to treat the patients. And we know sepsis can cause damage to vasculature and organs as well. So one approach to solving sepsis, to treating sepsis, is actually through extracorporeal therapy. So extracorporeal means bringing out the blood out of the patients and then flowing through a device and then cleaning the blood before you rub the blood back to the patients. So that's corporeal, extracorporeal therapy. And so when we were looking at this, we felt that maybe we can also try to see if we can develop a device to be able to detect and isolate and then to remove the bacteria. So again, we will use the cell margination effect where the red blood cell will actually flow through a channel, and then we will see if the stiffer bacteria would again get displaced to the wall of the channel, and thereafter we will remove them. We know that it works for the leukocytes, and earlier on I showed that you can work with the infected red blood cells, but what about bacteria? So again, we do an AFM imaging, and this AFM movie shows you a bacterial, even we can even see the pili and flagella. And we know the bacterial cell is a lot more stiffer than blood cells. And also it has a very unique elongated shape, about 1 to 2 microns. And we just wanted to see if this works. So again, we developed this microfluidic channel where we will have 2 stages of removal. The first stage is where the blood containing bacteria will flow through, and as it flows through the first channel, some of the bacteria will get marginated to the side. So when you get marginated to the side, you'll go into the waste. Then there's a second stage where we will further remove any leftover bacteria from the first stage. And again, those bacteria will again marginate to the side and move to the waste. And then thereafter, we should be able to get filtered clean blood. So this is our approach to see if we can remove bacteria from blood. So let me show you what happens. So we stain the bacteria, and in the first channel over here is where we input the blood with the bacteria. So when we first input the blood with the bacteria, you can see the bacteria is all over the place. It's brightly littered, so you can see there's no margination. So as it goes through the first channel, this is what happens. You can see that now there's some of the bacteria actually gets marginated to the side. There's still some bacteria in there. So this is the first bifurcation. At the second bifurcation, where we had the second stage separation, this is what happens. So you can see that some more bacteria gets marginated to the side, and there's still some bacteria left, but the amount of bacteria left is actually a lot more lower. So so there's a decrease in the amount of bacteria that passes through at the second stage. So over here is filter blood with a smaller, lower amount of bacteria as compared to in the waste. So we also tried yeast as well, and we found that we are able to also remove yeast cells from blood as well. But in general, we are able to, for this case, remove about 80% of the microbes of the bacteria. So using this device, we are able to remove quite substantial amount of this bacteria. Of course, we can again multiplex the system and then redo cleaning if we need to. So we could in effect have not only 2-stage but also 3-stage to remove even further bacteria if we need to. But for this 2-stage system, we are able to remove more than 80% of the bacteria. But of course, the system itself is very low throughput, so we need to find ways to do more high throughput testing. So because I mean, a human has 5 liters of blood in our body, and so we need to be able to process and remove blood in a very high throughput way. So one way is to multiplex. For example, this is just to proof of concept to show you that we can actually be able to do a 6-channel parallel system to remove blood in parallel. So right now I'm just showing you 6 channels, but we can literally have 100 or even a few 100 channels to increase the throughput of this removal. So in fact, we even show that we not only can do parallel, but also we stack the number of microfluidic channels as well. So over here, we see a multiplexing of 18 channels. But as I mentioned, we can literally multiplex to many more channels if need be. So this is one device we developed to show that using differences in the mechanical properties of bacteria from healthy red blood cell, we are able to actually remove the bacteria from blood of patients. So the last one I want to talk about is cancer. We know that when someone develops cancer, the cell can actually undergo uncontrolled growth, and when that happens, it will develop into a tumor like this. If the cell remains where it is and doesn't undergo metastasis, then it's a benign tumor. So if the patient goes for surgery to remove the tumor, the patient will recover from it. However, the ability of the cancer to disseminate and form a new secondary tumor at distant site means that the cancer cell has become malignant. And in fact, metastasis is responsible for more than 90% of cancer death. And so it's a very important disease for us to look at and to understand the whole cancer metastasis process. So what happens is, if a cell becomes malignant, it is capable of detaching from a primary tumor. It is able to squeeze its way into the circulatory system, and it can move from one organ to the next organ. For example, from breast, it can move into bone, and then it can squeeze back to form secondary tumor like this. So there are a number of critical steps from escape from primary tumor to intravasation to circulation in the bloodstream, and then eventually extravasation, and then establishment of a secondary growth, and then thereafter initiation of angiogenesis to form a metastatic tumor like this. So this whole process is actually very mechanical because firstly, the cell will become more deformable. It will actually lose its adhesion, and thereafter it's able to detach from primary tumor and then squeeze its way into the circulatory system. And so we want to understand how this can enable the cell to undergo such metastatic cascade. So when we will, there are already literature showing some of the cells. For example, this is in vivo imaging of cancer cells undergoing very large deformation in capillaries of mice. And you can see that some of the cells literally have to undergo large deformation in capillaries. And you can see the nucleus itself also plays a very important role because they get excessively deformed. And yet, despite this large deformation, the cell will still be able to traverse through all these capillaries to reach to its final destination to form secondary tumor. So it means that the role of cell mechanobiology is actually very important in metastasis. And so there are two parts to it. What constitutes a cell mechanical properties? One is actually cytoskeletal, the cytoskeleton itself, which leads to the cytoskeletal mechanics. And secondly is the nucleus itself. So nuclear structure, of course, will determine nuclear mechanical properties, and both cytoskeletal and nuclear mechanics will actually determine how cell mechanobiology, in the sense how cancer cell will respond in its external environment and be able to further undergo metastasis. So in our lab, we actually perform many different tests. I don't have the time to go through all of them. We did single cell nuclear mechanics to cell adhesion, to looking at cancer cell migration, and even looking at chemomechanics of anti-cancer drugs. I'll just focus on the single cell nuclear mechanics and then show you some of the devices or technology we developed to detect cancer. So one of the interesting experiments when we first go into this area is to perform looking at deformability of cancer cells using microfluidic channel. So this is MCF10A, which is a non-cancer cell. If you look at the cell itself, as it goes through a 10 by 10 micron channel, it takes a really long time to go through a channel like this. For a metastatic MCF7, which is a breast cancer cell, if you look at the cell itself, the time taken to traverse through the channel is much faster compared to the MCF10A. For the highly metastatic MDA, MB231 cells, the time taken is even faster. So you can see that, again, benign cell takes a longer time. Metastatic MCF7 takes a slightly faster time. And then the highly metastatic cells is even more faster. So it shows that actually the malignant cells are much more deformable. And of course, we also did look at the nucleus deformation. So if you look at this video here, you can see the large deformation on nucleus undergoing deformation through a very narrow channel like this. So nuclear mechanics is also very important, the mechanical properties. And of course, we did quite a number of different experiments, both looking also at the doing confocal imaging to look at the cytoskeletal structures of cancer cells. So this is MCF10A, again, non-cancer cells. You can see very well formed F actins within the cells as compared to MCF7, where the actins are a bit more disorganized and not so filamentous. Of course, we also look at nucleus as well, where for the non-cancer cells and versus the MCF7 cells, the lamine AC structures are much more well-defined and a lot more expressed in the non-cancer cells versus the MCF7 cells. So that shows also why the non-cancer cells actually, non-cancer breast cells takes a longer time to actually squeeze through a narrow channel. I mean, if you were to look at how the nucleus actually deforms and occupies the whole channel, this is also one contributing factor to it being much more stiffer than the cancer cells. So we did quite a bit of these different studies, and then when we were talking to clinicians, that they actually was very interested to see how we can use our understanding of this cell mechanics to see if we can use it to detect and diagnose diseases, diagnose cancer. So we started to look at how clinicians do cancer diagnosis. So one way they do it is to tumor biopsies, where you take a sharp needle, you poke through the body to sample cancer cells from the patients. So we know this procedure is very invasive. It's very painful. You cannot do it frequently. Normally, do it once before chemotherapy treatment and once after chemotherapy treatment. And so in the course when the patient is undergoing treatment, there's no way for us to sample cancer cells to see if, for example, the tumor has shrunk or what has happened to cancer cell. Have they been destroyed by the anti-cancer drugs? So, but we also know, as I pointed out just now, that cancer cells do metastasize through the circulatory system. So is it possible for us to actually be able to capture cancer cells from blood, which is what we call liquid biopsies, rather than from tumor biopsies? So the idea is that if you can do liquid biopsies, then they can complement tumor biopsies as well, and hopefully change the way cancer is being diagnosed and treated. Because if you can do liquid biopsies, it's non-invasive, it's not so painful, and in fact, you can do it quite regularly. You can do it every 2 weeks or every month to track the status of the patient with regard to the metastatic cancer cell that's circulating in the human body. So in fact, liquid biopsy itself actually is quite a hot area. When we first started this research about 15 years ago, it was still very new. But in the last 5 years, there's a lot of work now being done to perform liquid biopsies, not just capturing cells, but also the cell-free DNA associated with cancer. And people now also looking at exosomes as well. It's a game changer because if this really works, then this will become kind of a standard test that we can actually do in a clinic or hospital very easily, because it's just doing a blood draw, and from it, if we can isolate the cancer cells, then we can analyze those cancer cells. So liquid biopsies, I would think maybe hopefully in the next 5 to 10 years will become a standard test, because I think a lot now data are coming up from this work, and I think it's pointing to the fact that now we are able to actually be able to, for example, determine mutation in patients from such a test, a liquid biopsy test. But of course, it has a lot of challenges if you're trying liquid biopsies, because for example, if you try to isolate the cancer cells, or we call it circulating tumor cells, it's actually very challenging, because in blood we have 40 to 45% hematocrit, but cancer cells are very rare. In 1 ml of blood, there are 5 million red cells, 8 million white cells, but there are about less than 10 to over 100 cancer cells. And what makes it even more challenging is that cancer cells are very heterogeneous. Some are epithelial, some are undergoing epithelial mesenchymal transition, some are actually cancer stem cells. And so if you were to use antibody to capture them, then you may only capture a subpopulation of cancer cells, but not all the cancer cells, because they all express very different proteins on the surface of the cancer cells. So for us, we try to do away with using antibodies, but instead use the difference in mechanical properties of cancer cells from blood cells to isolate the cancer cells. So we developed a suite of microfluidic chips to do that. There are 4 of them, but I'll just show 2. One is the microfiltration, and second is the spiral. So in the microfiltration, what we try to do is to be able to physically trap and isolate the larger and stiffer cancer cells from blood cells. And this is how the chip looks like. It's a PDMS chip that's mounted on a glass slide. And inside the chip are these semicircular traps that can actually trap the cancer cells. So how it works is inside the chip are these 3 micropillars that's sticking out. And if you look at this figure here, movie here, you can see that the traps are some of them are tilted slightly to the left, some are tilted slightly to the right, and they are staggered. And in this chip, there are about 14,000 of these microtraps. And in these 3 pillars itself, they're placed about 8 microns apart. So if there's a cancer cell that's larger and stiffer, you'll get trapped like this. Whereas the red blood cells and white blood cells being smaller and more deformable, they'll be able to squeeze through these pillars. So from this movie here, you can see that there are cells that get trapped like this. And so from this device, we're able to trap the cancer cells as we flow the blood through this device. And so again, just show you how it works. When the cancer cells come and encounter 3 pillars like this, it's stiffer, it will get trapped. Then once the cells are all trapped, what we can do is we can then reverse the flow to isolate the cancer cells. So over here, to release the cancer cells, we can reverse the flow, and then we are able to release and then be able to retrieve the cancer cells, and thereafter we can do the analysis. Just to show you processing of actual patient blood, this movie shows you the huge amount of blood that we are processing from patients. You can see there are so many red blood cells, but I will show you a close-up movie of some of the cancer cells that get trapped. So again, in this movie, there are all the red cells and white cells flowing through, but if you look very carefully, among the 3 pillars here, there's actually a cancer cell over here. There's also another cancer cell here. So this movie shows you that despite the fact that there's so much cells flowing through this, we are still able to capture the stiffer cancer cells using these 3 micropillars. When we were doing this, actually, this device took us about, I think about 7.5 ml of blood, it took us about 3 hours to process it. So we found 3 hours is quite long. So we started to look at another different microfluidic design to isolate cancer cells. So this is the the device that we come up with, it's actually a spiral channel. What happens is we input blood over here, and as the blood flows through a curved channel like this, and as it comes up, what happens is the larger cancer cell will get focused near the wall of the channel. The smaller red blood cells and white blood cells will get focused away from the channel. So through just flowing through a curved channel, we are able to separate the larger cells from the smaller cells. Of course, for this device, then we can only work with cancer cells that are much larger than blood cells and white cells. So how does it work? Actually, it's based on the principle of inertial focusing, where if the cell is flowing through a straight channel like this, you will undergo two types of forces. One is called the shear-induced lift force that will push the cell near the wall of the channel. The second is the wall-induced lift force that will push the cell away from the wall of the channel. So what happens is in a straight channel, as the cells experience Poisson flow, it will actually it will reach an equilibrium position where the cell is somewhat a bit away, some distance away from the wall of the channel. But if the channel is a curved channel, then what happens is there will be a centrifugal force which will push the fluid from the inner wall to the outer wall. So it's like spinning in a washing machine. The clothes get pulled outwards. So when that happens, it will create what we call a counter-rotating Dean vortices, and this counter-rotating Dean vortices will create a Dean drag force. So if you look at this equation here, so the equilibration of the different size cells will depend on the lift force over the drag force. And it varies with the diameter of the cell to the power 3, which means that if the cell size is larger, the lift force is higher, it will pull the larger cells nearer the wall of the channel. If the cell size is smaller, then the lift force is actually lower. So the drag force will then pull the cell away from the wall of the channel. So as it comes out, we can then be able to equilibrate the cells of different sizes at different locations. So using this principle, we came up with this microfluidic channel to separate the cells, the larger cells from the smaller white cells and red blood cells. So through this very simple device, by flowing through a spiral channel, we are able to separate the cancer cells from the blood cells itself. So when we developed this, there was a lot of interest in this. So we thought that we would set up a spin-off company to commercialize this. So we have a company now called Biomedical. And this is the system that we have developed, where we have the spiral chip that's enclosed in the cartridge. It's mounted on the top. And then what we do is we can collect 7.5 ml of blood from the patient, plug it over here, press a button, and thereafter about within 30 minutes to 45 minutes, we can collect the cancer cells from the other tube. So it's called a clear cell FX system. We have actually obtained our CE-IVD mark as well as China and US FDA listing as well. So currently it's installed in 90 over locations around the world. So right now we are actually, some of the labs are actually using this. We have actually conducted more than 3,000 over clinical samples for 3 types of cancer: breast, lung, and also colorectal. So ideal for this system is eventually for targeted therapy or precision medicine. What we do is we can collect blood from patients, use our system to isolate the cancer cells, and then thereafter we can do genetic and molecular analysis. And with this information, we can put in the report. One of the critical information we need is actually what we call druggable mutation. If a certain mutation that we spotted is treatable by drug, then we can administer the right drug to the right patient. So this enables what we call personalized treatment or precision therapy. Moving forward, actually right now we are looking at having an integrative approach to precision medicine. So we not only develop microfluidics and using this mechanobiological principles to separate cancer cells as well as exosomes as well, but we also now performing quite a number of omics like genomic, transcriptomics, proteomics, and metabolomics as well, analysis. And the last 2-3 years, actually, we've been doing quite a fair amount of work in terms of data and analytics. With this huge information we gather, we are trying to see if we can use data analytics for us to come up with a better panel of biomarkers for us to better diagnose as well as prognose cancer as well. So we have actually a number of papers published in this area. I don't have time to go through them, but mainly we came up with what we call 29 ECM gene-related panel for us to better prognose and diagnose lung cancer. And this relates to the extracellular matrix genes. So with that, I'd like to make some concluding remarks. So I've actually show you some of the disease mechanobiology research that we conducted. Those are very basic research, but I think conducting this disease mechanobiology research is quite important because they actually can give us a different perspective of diseases and help us to better understand the pathology of diseases that clinicians and biologists normally would not have done, because their approach is somewhat different from what we are trying to do here. The other thing is what we observe is that the altered cellular mechanical properties that we have observed can actually act as biomarkers, not only for us to do disease detection, but also diagnosis as well. And of course, there's also potential in use of the knowledge that we develop here in trying to develop therapeutic strategies to inhibit pathological outcomes. For example, perhaps if we can tune, for example, cancer cells to have properties that are closer to its healthy counterparts, that might be one way for us to come up with a therapeutic treatment for cancer patients. So this is there's a potential there, and I think it's something that we are very interested to go into. So to bring it into perspective, we did disease mechanobiology, and from here we can bring some insights into mechanopathology. But at the same time, with this knowledge, we not only can come up with bio-inspired diagnostics and therapeutics, but moving forward also, hopefully the knowledge we gain can also help us to develop what we call mechanobiomedicine or treatment of some of these patients suffering from some of these diseases. So before I end, I just want to acknowledge a number of people, the engineers and researchers and students. For malaria, for example, we'll be working with Professor Subha Suresh, who is now the president of NTU, and also Jay Han from MIT, and our postdocs, Majid, Rilwan, Sujin, Ali as well as Subin and Bina. We also work a lot with the clinicians from both the National Cancer Center and also National University Hospital, and also Cancer Science Institute. And although I'm an engineer, but I felt that it's very important also to understand biology. So we do actually collaborate a lot with the biologists and try to also learn from them, like John Paul Thierry and Kevin. John Paul is actually working in cancer and Kevin in malaria, and Professor Usa from Mahidol University has been helping us actually with our malaria project, especially in obtaining some of the clinical samples. And of course, because we are very interested in spinning out some of our devices and technology, so we do work with the industrialists and investors as well. For example, with Manchun, who's working with Biomedics. And of course, I want to also thank the patients. Actually, we don't know who the patients are, but they are very generous in giving us their blood for us to do the tests. And some of them actually are very sick. And so we are very thankful to them for their contributions. And of course, I want to thank the funding agencies as well, NBI, Spring, SMART, and also National Research Foundation. So before I end, I just want to make a quick announcement that we'll be organizing the World Congress of Medical Physics and Biomedical Engineering next year, 30th May to 4th June. So hopefully by then, this pandemic will be over, but we'll see what happens. But this World Congress is held once every 3 years. So it will rotate in North America, Europe, and Asia. So next year it will be in Singapore, and actually we'll be chairing it. So we are expecting about 2,500 delegates. So you are welcome to actually join us. If you want to find out more, you can actually visit our website at wc2021.org. And finally, in this pandemic situation, hopefully you'll stay safe and healthy, keep your distance, wash your hand, and don't touch your face. Also wear masks, which is really what we are doing right now here in Singapore as well. So with that, thank you so much for your attention, and I'll be happy to take questions.
Thank you very much, Professor Lin, for a talk with not only insight in fundamental science, but also real-world application, particularly in the clinics. And it's my understanding that you're also working to fight the COVID-19. Maybe next time when you give a talk to us, you'll tell us about that. Before the question and answer period, let me do a short ad of our next presentation, which will take place Saturday, July the 25th at 10 p.m. Beijing time, exactly this time 2 weeks from now. And Paul Ying will tell us about healthcare fulfillment in the emerging economies, foreseeing the future development of biomedical engineering. So very applied topic. People who are interested in this topic should mark it on their calendar and join us in 2 weeks' time. So now the floor is open for questions. I will select questions based on their relevance. I see quite a few people have raised questions, but some of them are not entirely relevant. In the meantime, if your questions didn't get answered, you can email Professor Lim in the email outlined in here. And we also would like to thank Columbia BME for hosting the talk. So the first question is the process of filtering red blood cells in the multiplex channels traumatic?
Right, yeah. So this is a very interesting and good question. Yeah. So when we filter the blood, we do take note of the forces that's acting on the red blood cell. So when we designed the microfluidic device, for example, we would carefully kind of calculate what the shear forces and pressure will be acting on the blood cells. Of course, when we do the experiments, after we collect the RBCs, we also look at whether RBC has been lysed and so on. So the filtering actually, I would say, is not traumatic in the sense that we want to preserve the red blood cell after flowing through the device. So it could be, for example, either slowing the flow or designing the structure such that it will not create any effect that would damage the red blood cell. So I would say that this is part of the design consideration.
Thank you. Next question. Since cellular biomechanics may be influenced by extracellular mechanical signals, how can we relate properties in vitro to properties in vivo? Thank you.
Right, yeah. So that is okay, another interesting but not easy to answer question. Yeah, I mean, a lot of experiments we do are yes, definitely in vitro. If you talk about in vivo, right, it's hard to actually be able to estimate, for example, the kind of for example, if you're talking about in the tumor itself, right? When we one of the research we have is looking at cell adhering to a tumor and looking at the tumor stiffness and whether the difference in the tumor stiffness will either encourage or discourage cell proliferation or tumor growth. And most of the time, it's not that easy to know exactly how much of the forces or pressure is actually acting within in vivo itself. I think the only closest we can do is maybe in the mice where we can insert certain probes to kind of measure what the forces is. But certainly in the human body, I don't think we can actually be able to actually do that. We can estimate, but we will not be able to do that. But we will also try to see, for example, in mice, we can probably be able to probe it and then try to mimic the kind of parameters that what we think we are observing in vivo to the in vitro environment. Yeah, so it's not that easy if you want to say have an in vitro that will be able to perfectly mimic in vivo properties.
Thank you. Next question. Can you please give some explanation on the reason why malaria infection increases cell stiffness? Is there any molecular understanding of how the infection increases the stiffness?
Right. Yeah. So there are two parts to it. When malaria when the malaria invades into the red blood cell, it will actually causes changes. So it actually, firstly, it will multiply. it will consume the hemoglobin. It will actually produce many byproducts within the red blood cell. And so some of these additional products in the red blood cell will cause the whole red blood cell to be stiffer. And then secondly is that it will actually export protein to the membrane. And some of this protein will interact with the spectrin network, which will cause the spectrin network to be also stiffer. And so when we do like micropipette aspiration of the, to measure the shear modulus of the membrane, we'll see also there's an increase in stiffening of the membrane. So there are two parts to it. One is within the cell, because of this byproduct that's produced when the parasite consumes the hemoglobin, that causes the whole cell to be stiff. And then second is the membrane itself. So that's what causes the whole malaria infected cell to be stiff and also sticky.
Thank you. As time is drawing very close to the end, I would like to maybe ask the final question. So you mentioned the clear cell, the commercialized product can work with breast, lung, and colorectal cancer. I didn't hear hepatic cancer, which affects the Asian population in particular. Is that in progress, or is that something that's already done, but you didn't mention?
Yeah, so hepatitis, we have actually also tried. We were able to actually, at least for the small number of tests that we've done, we found that we were also able to detect them. The reason why we're going for like breast and lung is also because those are, because when you go for FDA approval, normally you have to state, what type of cancer you're going for. You cannot say all cancer types. And so we are kind of zooming in on the more serious cancers, more prevalent ones. So lung, breast, colorectal, actually also prostate, for example, are the cancers that we're looking at. Okay.
Well, there's one final question which just came up, which I think is important. Maybe you can briefly answer this. Is it possible to remove cancer cells from blood for those patients with late stage cancer that are too late for surgery?
Right, yeah.
Blood cancer.
Yeah, so that's a good question. And in fact, I didn't mention, but we did develop, we kind of multiplex and stack up a number of microfluidic chips to do cleansing of cancer cells from, cancer cells from blood cell, right? So it's like a dialysis system.
Yes.
But the reason why we do this is because we're talking to a clinician. The clinician was doing operation on the spinal tumor of patients, and when they do operation, the patient tend to lose a lot of blood, sometimes 1 to 2 liters or even more. And he was asking, can we salvage this lost blood and then clean it of the cancer cells before they route the blood back to the patient? So in that context, we came up with a system to clean blood of the cancer cells before we route the blood back to the patient instead of using donor's blood. But if you're talking about trying to remove cancer cells from blood for very late stage patients who cannot go for surgery, then we are not treating the cancer at the source, because the tumor will still be there. The tumor will still be shedding cancer cells, and even if you can remove cancer cells from blood, the tumor will still be there. So normally we don't treat patients that way. Yeah.
I think there's a subsequent question about blood cancers. Would it be possible, viable strategy for removing the cancer cells for blood cancers?
Right, yeah. So it's a good question. As I mentioned, because we are trying to remove cancer cells, because they are stiffer, they are basically larger or stiffer than blood cells, right? So if the blood cells, blood-related cancer cells, then it's a bit difficult. But having said that, actually right now I have a project looking at myeloma, where we are trying to also remove some of these cancer cells, but it's a different approach then. Yeah.
Okay, thank you very much. I'm afraid that's all the time we have. Once again, if you have more questions for Professor Lim, you can email him on the email address that I have on my screen. I'd like to thank Professor Lim one more time, and thank you all for attending, and thank the Department of Biomedical Engineering at Columbia University. Make sure you register for our next webinar on July the 25th. Enjoy the rest of your weekend. Thank you very much.
Thank you. Bye.
Thank you. Bye.
Thank you, CT. Yeah.
Thank you.
Thank you, Chwee. Fantastic job. Thanks, CT, for fantastic talk.
Thanks. Are we offline already?
No, not offline, so
Okay.
you know, but people are probably logging off. But
Okay.
Thank you again for the
All right.
invitation. Yeah.
Well, thank you very much. That was a great talk. I really enjoyed it.
Yeah.
Oh, thank you. Yeah.
See you in 2 weeks. See you in 2 weeks.
Yes.
2 weeks.
Bye-bye.
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 Bye-bye.